Okay, so for the second talk, we have the pleasure to listen to Gabriel Amselem from École Polytechnique, who will talk about growing from a few cells to a population and back. Please, Gabriel.
Hi. Thanks a lot, Aline. So thanks a lot for organizing this this workshop. It's very interesting for me, and I have to say there is a variety of topics that I'm honestly not that familiar with because I'm not a mathematician, I'm not a statistician, I'm not a biologist. I work at LADIX, which is the lab for fluid mechanics at École Polytechnique. So I specialize in microfluidics, which is fluid mechanics at a small scale. And what I'm going to show you is how we designed a device that allowed us to monitor the growth of populations of cells, of bacteria, over time on hundreds of populations. And we observed that the populations were very heterogeneous in numbers, okay? And that led to two questions. The first one is, can we understand this heterogeneity in population sizes, starting from the heterogeneity of division times at the single cell level? And the second question is the end back. So the second question is, once we know this heterogeneity in a population sizes, can we go back to the heterogeneity at the single cell level? And this is really the work mainly of Antoine Varisien, who was an excellent PhD student co-supervised by Charles Barraud and myself between LADIX and Institut Pasteur. And we had a collaboration which was crucial with people that many of you know from at the CEMAP at Polytechnique, so Tristan Roget, Aline Marguet that everybody knows here, Vincent Bansay, and Sylvie Mellea. So bacteria are prokaryotes. They are very simple cells. They don't have any nuclei, and they're everywhere around us. If you take a spoonful of water, you're going to find anything from 100,000 to 1 million prokaryotes. If you take a gram of soil, you're going to find 1 million to 1 billion prokaryotes. And you don't even have to look around you. You can just look at yourself. You probably heard of the gut microbiota. So we have in our guts 10 to the power 14 bacteria, which is incidentally 10 times as many bacterial cells than we have of our own human cells. Okay, we have more bacteria cells than human cells in our body. How do we grow these bacteria in the lab? Well, we have two different ways of growing them. One possibility is to grow them in a petri dish, so on a solid culture. This is an agar here, petri dish. So it's a hydrogel that has nutrients. And if you put a source of bacteria, for example, if you stick your hand in the petri dish and you wait a couple of days, then this is what you get. You see all the bacteria growing, and there's also fungi in there, right? As colonies, okay? So bacteria are really everywhere also on your hand. And that's one way of growing them. The second way of growing them is to take a sample and not put it on a solid medium, but on a liquid medium. So you take your sample from your petri dish or from anything you want, and you put it in a liquid culture here. So that's nutrients for them. And you wait, and what you see is that the liquid culture becomes turbid because you have bacteria in there. So they make it turbid. And by looking at the turbidity of the medium, you're able to determine how many bacteria there are in there. Okay, and that's very well known. I'm not inventing anything here. You get these very stereotypical growth curves of bacteria that were already depicted in a review by Monod in 1949. Here on the y-axis, you have the logarithm of the number of bacteria. On the x-axis, you have time. And what you see is essentially 4 phases. So at first, you have what people call the lag phase, where apparently nothing seems to grow. So people say that bacteria adapt to their environment here. Then you have an exponential phase here. So that's a log-link scale, right? So that's exponential growth. With a growth rate alpha. Then you have a saturation, where bacteria stop growing and then bacteria die. And as an aside, going back to what Sylvain Billard was saying last week, right? If you look at this macroscopic curve, you actually don't know what's happening at the microscopic level. So for example, if you look at saturation, it could be that cells just stop growing, or it could be that you have as many cells dividing than cells dying. And you see a saturation. You have no way of knowing this just looking at this curve. But this is the stereotypical growth curve of bacteria. And we can do this in the lab. These are the growth curves of Antoine Varisien. You have the turbidity of the medium here, which is related to the number of bacteria in the medium as a function of time. and you repeat the experiment 3 times, and what you see is that it's very reproducible. You have some kind of a lag phase here. This is an exponential phase, and then you have some kind of a slowing down of growth and saturation. But the key thing here is that it's very reproducible. And now the question is, can we reproduce this experiment, not 3 times, but dozens of times, hundreds of times, and see what's happening? And that's where microfluidics comes in. So what we designed is a device that looks like this. It's a channel, okay, that you see in blue because we flowed in blue water in it. You have 2 inlets, 1 outlet. And if you look at the device under the microscope, you see these small squares here. These small squares, when you look at it from the bottom, are actually small holes in the channel ceiling. So if you look at it from the side, your channel looks like this. You have the glass slide here, you have your channel with these small pockets here, these small holes, okay? And above you have the material that composes your device, PDMS. That's very useful for us because now what we're going to do is we're going to put droplets in each of these small holes, the droplets of nutrients of liquid medium with bacteria in them. And these droplets are separated by oil, so the droplets do not interact. So we really have, you know, on a single device like this, we have 1,500 droplets. So that's 1,500 test tubes in which we can monitor the growth of populations. Okay, it looks like this. If you, from the beginning, it looks like this. So we have here our small pockets. We look at it from the bottom. Here, everything is filled with a medium that contains bacteria, which you do not see because the magnification is too low. And you push this medium with oil, and you see the oil-water interface arriving. And you see that this thing self-digitizes into small droplets. And so you produce like this, 1,500 droplets in a couple of minutes, roughly on your device. And then you have a look under the microscope at all of these droplets. Not all of them, but several hundreds of them. So this is what it looks like. We are able to scan 900 droplets per chip just because of time constraints, because the microscope has to move the sample. We're growing Bacillus subtilis, and you have an example of droplets here where what you see is bacteria growing. So the bacteria are these dark dots, dark spots. And the bacteria are fluorescent, which is very nice for us because using the fluorescence of the droplet, we're able to estimate the number of cells per droplet. So we're using the fluorescence as a proxy for the number of cells per droplet. Okay. So we can measure the fluorescence of hundreds of droplets in the experiment of about 900 droplets. Some droplets are empty, it's shown here. And each of these green curves is a different droplet, so different population that grows as a function of time. The black line here is the average population size, and otherwise you have all your scattering in population sizes here, right? And if you go to log-ln scale, then what you see is that you indeed have this exponential growth in this region here. Before that, you're in the experimental noise, where we don't detect very much what's happening inside the droplet. And above that, you're saturating. So bacteria have grown and they have used all the food that they could use. And the question we asked, as I told you at the beginning, is why do we have this spread? And more precisely, can we quantify this spread and relate it to parameters of single cell division? So to do this, we need a model. And as Alex told you before, cells fragment. So we have a fragment fermentation model. And there are 3 different kinds of models in the literature for cell division. The first model is what's called the timer model, where cells elongate. And after a given time, after 20 minutes, plus or minus 5 minutes, they divide. Okay. Now, this model is clearly not the good model. Why? Because we know that in a population of cells, the size of all the cells is roughly the same. Whereas if you have a timer model, you're going to have a huge distribution of sizes. Okay, so that's not the real biological model that corresponds to reality. It's a model. The second model is to say, well, we know that the size is conserved in the population. So what the cells do is that they elongate, and then when they reach a given size, they divide. So that's called the sizer model. And the third model is called the adder model, where cells elongate, and once they have added a certain mass to themselves, then they divide. Okay. So you have these three different things, and there's a beautiful article in 2015 that showed that, at least for E. coli and Bacillus subtilis, the correct model is an adder model. So the cells grow, they elongate, and once they have added a given size to themselves, then they divide. The problem is that this adder model is analytically very hard, from what I understand, to work with. And honestly, if you look, although there are differences at the single cell level on bacterial size of these models, if you look at the number of cells as a function of time for these three models, they're actually very similar. So although the biological basis is very different, if you look at the population of cells that is described by a timer model, sizer model, or adder model, this is the distribution of population sizes as a function of time. You don't see much difference for all practical purposes between the three models. In all cases, you have an average population size that grows exponentially with time. And in all cases, you have a standard deviation of the population size that grows exponentially with time. Okay. So for all practical purposes, we are going to use a timer model, and we're going to base ourselves on this paper by Bellman and Harris, 1952, where they describe these branching processes, age-dependent branching processes. So what we're going to say is that at the single cell level, the division time is normally distributed with a mean tau 0 and a variance sigma, a standard deviation sigma. And we're going to define the microscopic coefficient of variation, sigma over tau 0. Okay, so this is going to come up a lot in the next slides. So it's the microscopic coefficient of variation, sigma over tau 0. And what we observe at the population level is the variability in population sizes. So we can simulate, populations growing from single cells using this model. And what we see is that, well, the mean goes exponentially with time, the standard deviation too. And we define the macroscopic coefficient of variation as the standard deviation divided by the mean. Okay. And what Bellman and Harris tell us is that if we take this process here, all the moments of the distribution of the population sizes grow exponentially with time with the same growth rate alpha. And so we can have a look at this just by looking at the distribution, for example, of the population sizes as a function of time. If you rescale these population sizes by exponential alpha t, then we converge to a asymptotic shape. You'll see it soon. Okay, so after a couple divisions, we converge to an asymptotic shape. And this shape depends strongly on the microscopic parameters of the model. So depending on CD mu, if all cells divide at the same time, then you have a very peaked distribution of population sizes, and if CD mu is large, so if you have a very large variability in division times, then you have a more wide distribution of population sizes. So that's very nice for us, because we thought that it would enable to link the macroscopic variability to the microscopic one. Yeah, okay, sorry. So that's the result. So if we have this simple Bellman and Harris model, the macroscopic coefficient of variation is linearly related to the microscopic one. Yeah, that's very nice, because it means we can also do inference, right? If we know that the population size has a coefficient of variation of 0.2, then it means that the CD mu is 0.2. So we're very happy. We thought we were done. And here is where the experiments are. So we know experimentally that the CD mu is of order 0.2, and we know experimentally that the macroscopic variability is much higher. Okay, so we have a problem, and which means that we have introduced other sources of variability in our experiments, which I'm going to show you are twofold. The first thing is that I've told you there's an average one cell per droplet at the beginning, but the distribution of cells per droplet at the beginning actually follows a Poisson distribution because cells enter at random in droplets at the beginning. So that's what we observe experimentally. The blue lines, the blue bars are experimental curves, data, and the red is a fit with the Poisson distribution. So we have on average 1.6 cells per droplet, and some of the droplets are empty, and some have 1 cell, etc., 2 cells, 3 cells. So that introduces a source of noise in our data, variability. However, the shape of the distribution of the population sizes will still grow. All the moments of the distribution will grow exponentially with a rate alpha, and so we can still rescale our distribution of population sizes. And depending on the value of lambda and of CV mu, we're going to have different shapes of this distribution. So, you know, if we have, this is for on average 1.5 cells per droplet. If the CV mu is very small, then you really see in which droplet you had 1 cell at the beginning, 2 cells at the beginning, 3 cells at the beginning. And if your CV mu increases, then you're blurring out all the differences between the droplets because you have so much variability in the division times. And we can still calculate the macroscopic coefficient of variation as a function of the microscopic variability in division times, which is this Bellman-Harris term here. But now we're adding the Poisson noise variability on the top of that. And this is what we get. So the blue line here is the pure Bellman-Harris curve that I showed you earlier. And this is then simulation in stars, and red lines are theory for the case where we add a Poisson distribution of cells at the beginning with different values of lambda. So I realized that there is not lambda equals 1.5, but it's not that much higher than the purple line, and still we're still off, basically. Okay. And so here we wonder what was happening, and what's happening is that there is actually another source of variability which we cannot control, which is biological, which is that I told you that cells need to adapt to their environment. Okay, so these are movies that we took with bacteria growing on a solid culture, agar pad. So okay, you just see the bacteria growing. This is very similar to the movie that Sylvain Billard showed last week. And now we can track here precisely at what time which cells divided. And what we see is that the first generation divides roughly every hour with a large deviation, large standard deviation. The second generation divides faster with a smaller deviation. The third one faster, and fourth one you've converged basically to the stable distribution of division times. Okay, so because cells adapt to their environment because of a biological process, we have this evolution in the division times at different generations. And now if we can again incorporate this in the Bellman-Harris model, this is again the population size rescaled by exponential alpha t as a function of the microscopic variability of stable division times. And what you see here is that there is not much of a difference anymore between the curves, depending on CV mu. Why is that? Well, because again, what's happening at initial moments is important. And so if you have a large variability in division times at the beginning, you're blurring out all the differences that could be due afterwards to CV mu. Okay. And we have, so Antoine did the math. And so this is the expression for the coefficient of variation of the population sizes. It depends on the standard Bellman-Harris coefficient of variation. And you have this term here. Sigma is the standard deviation of division times in the stable state after 4 generations. Sigma 1 is the standard deviation of the first division time. And if you have more than one generation that divides at a different rate, you just need to multiply by the different exponential alpha squared, sigma 2 squared minus sigma squared times exponential alpha squared, sigma 1 squared minus sigma squared, etc., etc. Okay, so you're increasing the noise at each time step, and this is what the the curves of CVN versus CV mu look like. And again, what you see is you're flattening your curves right here. So it's actually going to be much harder to infer anything because a small error in the CVN translates into a large error in the CV mu here. Okay, so that's the expression. Now, if you combine Bellman-Harris process, Poisson distribution of cells at the beginning, and first division times, and you recognize all the terms. So this is Bellman-Harris multiplied by first division time variability times Poisson, and here you just have Poisson times division time variability, and then you just have Poisson here. Okay, and now we can compare this to experiments. So in experiments, this is the average population size as a function of time. The blue dots are experiments. At the beginning, we're in the noise. Then we have this exponential growth, and then we saturate. And we can compare quite well with simulation in theory. I should add that here there is actually a one fit coefficient, which we do not know a priori, which is that we measure the fluorescence of cells, not the number of cells. Okay, so we're basically fitting for the coefficient that relates fluorescence to number of cells. So what's the fluorescence of one cell? That's basically what we're fitting for here. But if we have this one fit coefficient for the mean, and we use the same fit coefficient for the standard deviation, we have a very good agreement between, again, experiment and theory. Again, you know, what's happening here is that you're in the experimental noise, and above you're saturating. And if you look at the CV value, so again, that's experiment in blue. You have a region at the beginning where you're in the experimental noise. Then you have a region where the CV is constant, and then it decreases because you're saturating your droplets. And the thick lines are the lines where we take into account all the noise that we can take into account. So the Poisson process and the 4 divisions that have different division times, 4 generations. And otherwise, you know, if we just take into account the Bellman-Harris noise, the CV would be here. If just the first division would be different, we would be here for the CV. If we had a Poisson process and just the first division, we still increase the noise, but we're not there yet. And basically, that's not too bad here. So that's the answer to the first question. Can we understand the variability? Yes, we can. Oh, okay, sorry. And that's one last thing is that we looked at the shape of the distribution, experimental shape of the distribution of population sizes, rescaled by exponential alpha t, and you see that there is sorry, let me put this back here. You'll see that there is a range of times during which this histogram superpose on each other, and that's again the time where we have exponential growth. Okay, so that's experimental data, and then we saturate and we lose this stability. And we can compare this to the simulation, and it's again not that bad. So what I've told you is that we've used the model that we know is biologically not relevant. We've used the timer model, but that gives good results for the population size. And the variability comes from 3 different sources. First, all cells don't divide at the same time, but they also need to adapt to their new environment, and we have a Poisson distribution of cells at the beginning. So we were able to go from the microscopic to the macroscopic. And the question we want to ask now is, can we go in the reverse direction? So knowing this macroscopic variability in population sizes, can we infer the microscopic variability in division times? And I've already hinted at this, but just using the shape of the distribution or the CV doesn't work. It would work if we were in the pure Bellman-Harris case, because we have a linear relationship between CV and CV mu with a slope that's about 1. But here, in the more realistic case of a Poisson distribution of cells and the large variation in division times at the beginning, the curve that relates CVN to CV mu is very flat. And so if we make a small experimental mistake on CVN here, this translates into a huge mistake on CV mu. So it's impossible for practical purposes to infer the division times of cells from this experimental curve of the population size. And what we need to do rather is to look at the dynamics of division. And so what we do is that we, this is a simulation of the number of cells in a colony as a function of time. And we look at what Antoine called the residuals. So the residuals are the number of cells at a time t plus delta t minus the number of cells that there should have been if there were no noise, which is the number of cells at t times exponential alpha delta t. Okay, so you look basically at the difference between your experimental curve and the pure exponential, and that's a residual. And you can look at this at different times. You have to choose your delta t correctly so that it works good. But basically, you take a division time roughly, and what you see is that as time goes by, so the residuals are normally distributed. You can, I think, formally prove this in the case where you have a Markov process, and for the Bellman-Harris process, I think there is also a sketch of a proof in Antoine's PhD thesis. So they're normally distributed, and the variance here increases with time, right? The variance of the residuals, so you go from 2 to 100 here. And if you look at the variance as a function of time, you actually see that it increases exponentially with time, and that it depends on this CV mu, so the variability of division times. So the higher the CV mu, the higher the variance of the residuals at a given time. So now we can have a look at what's happening experimentally. These are simulations. Experimentally, we can also look at the residuals. The blue is experimental data. The red is a Gaussian fit. We can again define a variance, and we can have a look at how this variance increases with time. And in theory, the variance should increase at the same exponential rate than the mean size of the population. Okay, that's what we saw. That's actually also provable, and that's what we also saw in the simulations. Which means that if we take the variance of the residuals and divide it by the mean fluorescence, we should get a straight line. Okay, and the value of the straight line depends on the CD mu. However, and that's the last thing that didn't, well, not the last thing, the second to last thing that didn't work in experiments. If you look at the variance of the residuals divided by the mean fluorescence, you actually get something that decreases experimentally. Okay, and it actually took several months to understand what was happening here. And what's happening is that we're not binning the data correctly. So what's happening in experiments is that we're looking at growth curves that are above a certain noise threshold for n, and below a certain threshold after which the curves saturate. Okay, so if you look at your residuals here at time t, at this point, you don't have that many curves to sample from, and not here either. And here you have tons of curves to sample from. And apparently that's what makes this estimation go wrong. Rather, what you should do is you should bin the residuals not as a function of time, but as a function horizontally. You should bin according to n. You should look at the residuals depending on the number of cells n here, and then things work more or less, and you'll see where's the more, where's the less. So the variance of the residual as a function of the number of cells is a linear function. That's what theory predicts us, that these are simulations. Simulation and theory agree. That's good. And then these are experiments. This is the residuals. Each point is a residual, and we can look at the variance of the residuals as a function of the fluorescence here. And we can now divide the variance at each fluorescence by the fluorescence. And this is what we get. And that's actually not too bad, because what we see here is that we have, this is the part of the curve where everything is noisy. Then we have some kind of stability here, right, where we have exponential growth. And then the curves, experimental curves saturate. That's not too bad. So by looking at this value here, we should be able to infer what the CV mu is by looking at this value there. And Antoine did simulations and showed that, you know, this value of this variance of the residuals divided by the number of cells is really only dependent on CV mu. So basically, if you have a pure Bellman-Harris process, you have the blue curve. And even if you add noise on the top of this, if you add a Poisson process at the beginning plus a variance of division times initially, then you get the purple curve. So basically, looking at we're getting rid of all the variability of the beginning because we're looking at the dynamics of the process in the exponential phase. And the problem is that okay, I think I took a slide, but basically the problem is that our experimental point is here. Okay, and here we really have a problem that we don't really know how to solve now. The problem comes from the following thing: is that we're looking at fluorescence and not number of cells directly. And we do not know precisely the coefficient that relates fluorescence to number of cells. I mean, we fitted one value of the proportionality coefficient in the first part, and that's what gives us this point here. But cells have different levels of fluorescence. We cannot see this proportion. We cannot see at this magnification what the fluorescence of a single cell is because it's hidden in the noise. And so we basically need to find another way to count the number of cells to be able to use this method of the residuals that seems that like it would give good results just from a theoretical and numerical point of view. But experimentally, we're not there yet. So that actually means for us doing experiments with a better microscope, a confocal microscope, or counting the cells, getting rid of the fluorescence and count the cells manually, and then we hope that we'll be able to see something. Okay, so that's the conclusion of the second part. So in theory, we can infer these division parameters, single cell division parameters from the population sizes, which is for us very nice. We do this by computing the residuals, and experimentally, it's still a work in progress. So that's it. Thank you all for your attention. If you have any questions, I'll be happy to take them.
Thank you very much, Gabriel, for this nice talk. So are there any questions for Gabriel?
Yes, if I can.
Yes, please.
Thanks for the talk. It was very interesting. You didn't say anything about since you're measuring fluorescence, you have an underlying Poisson process also.
Yeah.
And so that should contribute to your variability. So how do you estimate?
You're right. So we didn't really look at this sort of variability because I think that the variability that's even more important than this one is the variability of fluorescence between cells. Here you have a huge variability of fluorescence, but you're right. If we wanted to be fully complete, we should include this Poisson distribution also, which we don't take into account. We also have a problem that we have a background fluorescence, right? And there's always a question of how do you take into account this background fluorescence? You just subtract it. What do you do with it? We don't really know. But yeah.
The detection is done with a CCD chip, or?
Yeah, well, in this case, it's a CMOS, but yes, yeah, it's a camera, it's a CMOS camera. Yeah.
Okay. Thanks.
Any more questions?
Hello.
Yes.
Thank you for this talk. Can I just ask? So how are you measuring the residuals? So is this also coming from fluorescence?
Yeah. So we have experimental curves. I'm sorry, I don't have them here. But basically, well, I can go back to the first curves that I showed you. Let me just, I think my computer is bugging, but we have fluorescence curves as a function of time. And so it's here. it's here. Okay, so from these curves here, this is the fluorescence as a function of time for each droplet, and then we compute fluorescence at time t plus delta t minus what should have been the fluorescence at t plus delta t in the absence of noise.
So if there is some proportionality between the fluorescence and the number of cells, it is also appearing in the residuals, right? So is it because they're on different scales that is the problem? One of them is variance and one of them is squared and one of them is
that's exactly it. So
right, okay.
Yeah, that's exactly it. So this is the big problem, is that
I see, okay.
and this is the coefficient of proportionality in the fluorescence. If we could get rid of this, then we'll be very happy.
So I mean, stupid question, but if you just look at the standard deviation, you lose this, right? Yeah. But I guess then you have trouble with the theoretical computation, do you?
Yeah, I think that's the problem. Yeah.
Right. Okay.
Yeah. Okay. I think that's the idea.
Okay. Thank you.
Okay. I have one question on the adaptation to the environment, the film that you've shown. Is it the experiment that you did? Huh?
Yeah, it's Antoine actually, but yeah, it's Antoine's movie.
Okay. And so you observe that it's really interesting to see the distribution for the different generation. I guess that's a lot of time looking at the films. And,
yeah, so in this case it was manual tracking, so it was actually easier than to spend time to automate it, because we just did a couple of experiments but manually looked at each cell and counted.
Yes. And so you see only so it appears that at the fourth generation it had converged.
Yeah, I mean, so the problem is that after the fourth generation, it's exponential growth on the 2D surface, so we don't see the cells anymore, basically, or don't see them.
Okay.
What we think is that if you look at this, I mean, first and second are clearly different. Second and third are already, not that different, and third and fourth are also not that different. So we're saying, okay, after the fourth generation, it's probably converged to something stable.
Okay, and how many cells for those histograms? You remember?
I don't remember. No, I mean it's probably I mean, for the first generation, it's probably a dozen or something like this. I wouldn't think.
Oh, okay, a dozen. So not that much.
No, it's not that much. I mean, we can see here 1, 2, 3, 4, 5. Well, no, it's actually. Yeah, I don't know. I would have to look into Antoine's PhD thesis.
Okay.
I mean, it's not 100 for sure. It's like between 10 and 100.
And so maybe I got it wrong, but in your when you take this into account, I had the impression that you only consider a difference between the third generation and the fourth generation.
Yeah, so what happened is that the formula gets complicated if we put all the terms here. But
okay, that's what you said about multiplicating.
Yeah, but otherwise for this simulations, he really took into account all the different division times at all generations.
Okay. Yeah. And you tested with all the combination. I mean.
Yeah. So this is here basically. This is this. I mean, it's actually not that bad in terms of, if you just take into account the first division, it's already much better than if you don't take it into account with the Poisson process. But having the 4 divisions that are different allows you to gain a bit of variability and be closer to the experiment, basically. But already the important part is Poisson and first division, that's different.
And did you try more than 4?
No, because we didn't know what to put. And the other thing is that if you see that the variability comes from this exponential sigma 1 squared or sigma 2 squared minus sigma squared. And so we're getting closer and closer to sigma as the number of increases. So I mean, really the big contribution is the sigma 1 squared, which is much larger than sigma squared. And when we go to the third or fourth generation, I don't have the numbers in my head, but it's already much more stable, much more narrow. So yeah.
Okay, thank you. Any more questions for Gabriel?
Oh, I think there's something.
Yes.
How do you explain the adaptation of the cells after 2 or 3 generations? Well, honestly, I don't know. So I'm not a biologist, but I think what's happening is that the cells, when they come into a new medium, they need to put into place a whole machinery of proteins that are going to enable them to grow efficiently or grow well on a given medium. And so the daughter cells already have part of these machinery in place, basically, and so it's easier for them already to divide than for the first generation. I think that's the idea behind it, but I'm not that sure about myself. Yeah.
Okay. And so now your trouble, I would say, is with the to link precisely the fluorescence measurement with the number of cells.
Yeah,
Yes.
I mean, ideally, what we would like to do is to have a device that enables us to count directly the number of cells. I mean, it's certainly doable, but it depends on the instrument you're using, basically. We have some new data that we haven't analyzed yet using a confocal microscope, so it's more precise, and we hope to be able to count the number of cells more precisely. Another way would be to maybe do something like holography, where you can count in 3D the number of cells. I mean, we don't know, really, we don't know.
But maybe it's a silly question, but the OD, that gives you the number of cells, no? Or it's
so you need
also an approximation.
Yeah, it's also an approximation, and you need a calibration at one point.
Yeah, so that's the same issue.
And here, I mean, we try to do a calibration, but the problem is that if you take cells that grow in a culture, not in microfluidics, and that you know that you have exactly, I don't know, 100 cells per droplet that you're going to have, and you do the dilution, we have no idea why, but the fluorescence of the cells that were not cultured in microfluidics is different from the fluorescence of the cells that are cultured in microfluidics. So I mean, there's a whole biology thing with biology that is very noisy and that we don't want.
Yes. Okay, thank you.
Thanks.
So now there are some times for discussion. If